class, welcome to chapter 6. In this chapter we're going to talk about the chromosomal basis of sexual determination. And so as you can see here in this introductory image, we have a organism that is, or a chicken for that matter, that is half hen and half rooster. And so we're going to look at how this is possible and how chromosomes play a role in determining the sex of an organism. So we're going to start by looking at mammalian sex determination. And very early in development, there is bipotential in these organisms. And that means that to start, the precursors for both male and female genitalia are both present in all organisms. So you have the precursor for ovaries as well as the precursor for testes. And with this, we have the Mullerian duct, which is associated with females, and the Wolffian duct that is present in males. However, if the organism is XY, which is the chromosome makeup of males, the gonads will become the testes, and then the Wolffian duct will persist, while the Mullerian duct is it disappears, it's degenerated. And if the organism is XX, which is the chromosomal makeup in mammals for females, then the gonads will become the ovaries, then the Mullerian duct will persist, while the Wolffian duct is degenerated. And once this happens, hormones from the gonads will cause the external genitalia to develop either in the male direction into a penis and scrotum, or in the female direction into the clitoris and labia majora. So as you know from general biology and just your general knowledge from your experiences, mammalian chromosomal sex determination is where we have XY that become males and XX that become females. And because humans are diploid organisms and they have two of each chromosome, this results in a 50-50 ratio of females and males. So if we look at our Punnett square here, you have a 50-50 shot of inheriting a X or a Y from the sperm, and 100% shot at receiving an X from your mother. Therefore, this results in a 50% chance to become a male and a 50% chance to become a female. So what determines whether these gonads develop into testes or ovaries? Well, the key player in this is the presence of the SARI gene, SRY. And so this gene is located on the Y chromosome. It's not located on the X chromosome. So even in individuals that have 2 copies of the X chromosome and 1 copy of Y, such as Klinefelter's disease, you will still develop testes. So what happens is, if SARI is present, you can see it leads to this cascade where the cells with the expression of SF1 develop into AMH and testosterone producing cells, and eventually you have the Wolffian duct develop into the male internal genitalia, the epididymis and the vas deferens and the seminal vesicle, as well as the development of the penis, the prostate, and the scrotum. If the SARI gene is not present, then these genes that are responsible for the development of ovaries are not repressed, and you have the precursor cells develop into follicles and estrogen-producing cells, and that estrogen will lead to the development of the female internal genitalia, such as the uterus, the oviducts, the cervix, the upper vagina, as well as the external female genitalia, such as the labia, the clitoris, and the lower vagina. So here we're going to look at a transverse section, so we're looking at across as opposed to front on, as we looked at before, of this development of the gonads. And so here we have indifferent gonads, so they're not specified into testes or ovaries yet. And at this stage we have the Wolffian duct, and this genital ridge is important. So this is where the gonad cells will migrate to, or the germ cells, to develop into the gonads or the genitalia of the organism. Now at 6 weeks, we have the genital ridge is now occupied by both somatic cells, so non-sex cells, and germ cells, so they've migrated to the genital ridge. And as you can see, we have both the Mullerian duct and the Wolffian duct, so this is unspecified as male or female yet at 6 weeks. And at this point, this is kind of the last step before we decide, or the chromosomes decide, if it's to develop into a male or a female. Now at week 8, if there is a Y chromosome that contains that SARI gene, then we start to proceed towards testes development. So at this point, the sex cords start to move away from the cortical epithelium and develop into rete testes, so very early testes, and so they kind of isolate themselves from the epithelial tissue. And there are cells around the Mullerian duct that are called Sertoli cells, and what they do is they secrete this anti-Mullerian hormone that blocks formation of this duct, and so that duct is only needed in development of ovaries in females, and so we need to get rid of it. And so these anti-Mullerian hormones are secreted to degenerate it and then preserve the Wolffian duct. Now at 16 weeks, the testes are continuous with the rete testes and connect to the Wolffian duct through these efferent ducts. So we have the spermatogonium and the testes cord, so this is where sperm is produced and transferred to the efferent ducts. And the efferent ducts will turn into the vas efferens with the Wolffian duct. So these kind of develop and merge into what we know in mature humans as the vas deferens, and this connects the testes to the urethra to give kind of functionality to the male genitalia. Now let's take a look at ovarian development. So similar to testes at week 8, if there's no Y chromosome and there's no SARI gene, that allows for those ovary developing genes to be functional and to be expressed, and thus we have the cascade that develops into the ovaries. At this stage, the sex cords at the center of the developing gonad, so these sex cords, are degenerated, and that leaves sex cords on the surface of the gonad. So if we compare that to the previous slide, or a couple slides ago, where the testes are developing, it's kind of the opposite. So in the case of the testes, the sex cords on the epithelial surface of the cell degenerate, leaving kind of the internal mass. And in ovaries, it's opposite, where the internal sex cords degenerate, leaving only the developing germ cells, or the developing gonad cells on the external layer of the developing gonad. And at this point, the germ cells that have migrated to the gonads, those start to develop into ova. And so if you're a woman and you're told this, well, men should be told this too, but when you're born, you have all the eggs that you're ever going to produce. And so these germ cells develop into ova very early on, and those are your allotment for your entire life. Now at 20 weeks of female development, the Wolffian duct will continue to degenerate and dissociate, because that's only associated with male development. And coming in to surround the germ cells are these new cortical sex cords that will come in and develop into the ovarian follicles and surround those developing ovum. And eventually, a little further down the line, the Mullerian duct will develop into the fallopian tubes, the uterus, and the cervix. So now let's dig a little bit deeper into the genetics of how this works. And so remember, if you have an X and a Y, then you have the SARI gene. And if there's the SARI gene, SARI represses the ovary forming genes and leads to testes forming genes. And the way this works is that SARI and SF1 are expressed, and they start to express testes forming genes. SARI also increases the expression of SOX9, which can go and lead to more expression of itself, so it's kind of a positive feedback loop. SOX9 will increase the expression of FGF9, which also goes back and increases SOX9. So we're getting a very large abundance of SOX9 being expressed in these developing germ cells. And when that happens, we have all these testes forming genes that start to be expressed and leads to the development of testes. Now, how this interacts with females is that SOX9 is something that represses beta-catenin. And then if this is suppressed, there's no way that ovaries can form. So if we have SARI, it leads to the expression of SOX9, which prevents ovary formation through blocking this beta-catenin. Now, if there is no SARI present and we are an XX, a female, then there's no SARI to repress beta-catenin, and WNT4, another gene that is involved early in development, and you'll see throughout this semester or this quarter, is kind of prominent in development, that increases and interacts with beta-catenin to express ovary forming genes, as well as genes follistatin, important in the development of follicles, and we get the generation or creation of ovaries. So the real key players in this is if you have SARI, then that leads to SOX9, who works to prevent ovary generation and then leads to the expression of testes forming genes. So SARI and its interaction with SOX9 is kind of the master switch, which tells the gonads whether to develop into testes or ovaries. So how do we know that SARI plays this important role in developing testes and ovaries? Well, in normal males, XY individuals, we have the development of male genitalia, which we can physically see. And if we look at the expression of SARI on a gel, we see that SARI is present. And down here we just have control autosomal non-sex genes that are there to show us that our experiment worked. Now in XX individuals, which should develop into female, which we can observe, there is no SARI present. And we have that female development, and we have our control to show that there is actual DNA present. However, if we take mice and we have them as XX females, and all we do is add the SARI gene, we have the presence here depicted by this band. We get the development of XX mice that have male genitalia. So this just shows that SARI, if we add it to a female, it shouldn't be present, remember, because it's only on the Y chromosome. But if we add it transgenically, then we get the development of males. So this is kind of that master switch, as I alluded to in the previous slide, that controls the development of male or female genitalia. So remember, SARI is kind of the master switch, but it acts in accordance with SOX9. So SARI is only active for a few hours, where it activates SOX9. So further, kind of looking at that same experiment as we did in the last scenario with transgenic SARI gene, we can also look at transgenic SOX9 to kind of see its role in development. So in a wild-type male, wild-type meaning there's no transgenic or genetic manipulation of any type, we see that we can stain for SOX9 and see that it's present, as by this kind of dark blue streak here, in the developing genital ridge. And this is at 11.5 days. Now at 16.5 days, we see that AMH, which is anti-Mullerian hormone, is present. So that's getting rid of that Mullerian duct and allowing for the Wolffian duct to be prominent in male formation. And so in newborns, we can look at this kind of reproductive and urinary tract where we have a kidney and the adrenal gland on top of it. We have the formation of the bladder, and then over on the side here we have the formation of testes. And this is expected in developing male mice. And so if we look at the histology and the cells within the gonad, we see these developing testes cells. Now if we compare that to a female, a wild-type female, so there's no genetic manipulation of any kind, we see that SOX9 is not present. And remember, SOX9 is activated by SARI. So since females don't have a Y chromosome, there's no SARI and thus no SOX9. And this cascade between SOX9 and SARI also activates that AMH. And so there's no AMH, so we don't have any degeneration of that Mullerian duct. We still have the proper formation of the Mullerian duct, and the Wolffian duct is actually degenerated. Now if we look at the gonad morphology, again we have kidney and adrenal glands like we would expect in both males and females. We have a bladder just like we would in both males and females, but instead you can see we have the development of a fallopian tube with an ovary in the top. And if we were to look at the histology, we can see it's starkly different from the testes, and we actually have cells that are more representative of ovaries. Now, if we do transgenics, and so if we take a female and we transgenically introduce the SOX9 gene, which is not supposed to be there because there's no Y chromosome, but we put it in, we see that along the genital ridge we have expression of SOX9, which is expected because we put that in there purposefully, but that also leads to, at 16.5 days, the expression of AMH, which shouldn't be there because, again, no Y chromosome. And if we were to look at the morphology of the gonad, we have kidney and we have the adrenal gland like expected. We have the bladder, which is expected in both males and females. However, we don't have an ovary, and instead we are developing, similar to A on the left-hand side here, a testes. And if we're to look at the histology, we see that the cells in this developing gonad, this developing testes, is much more reminiscent of the testes in a control or wild type male mouse. And so SARI is the master switch, but as said at the beginning of this slide, it's only active for a few hours, and then SOX9 takes over and actually kind of leads the development. So this kind of shows the relationship between the last slide and this slide between SARI and SOX9 and how they both play a key role in the development of testes. So if we look at the expression of SARI, here we used GFP tagging. So we have this green fluorescent protein added to SARI. And as I said in the previous slide, it's only active for a few hours. So if we look at the expression of SARI in the genital ridge at 11 days, we see that there's a little bit of SARI expression indicated by these green dots, but not a lot. But at 11.5 days, we have a whole bunch. And then if we were to look at they didn't include this figure, but if we were to look at 12 and a half days, that expression would go back down. So it's just like a light switch. It's just on for just it's active for a second, and then it causes this cascade. So if we're to look at 12 days and change our focus from SARI to SOX9, we see that in the genital ridge at 12 days, SARI has been active just long enough to cause this very stark increase in expression of SOX9, which is responsible for suppressing the ovary generation and leading to testes formation. And because if you remember that figure, SOX9 had this arrow that pointed to itself, this kind of circular arrow, that means that SOX9 is able to increase or promote expression of itself. It activates its own promoter, which makes it present long term. So once SOX9 gets turned on, it's turned on for a long, long time, as opposed to SARI, which was only present for that short period of time. So if we're to look at 13.5 days here, we see that as these testes are developing, SOX9 is continuing to be present and continuing to push this development of the gonads towards becoming testes. One key milestone of development of testes is when there are cells called mesonephric endothelial cells that migrate from adjacent kidney tissues into the rudimentary gonads. And so these cells will move from the kidneys into the rudimentary gonads and form blood vessels. And so in this experiment here, to determine the role of SARI in this process, they took mice that were 12-day-old mice embryos, and they removed the urogenital rudiment and the gonads. And so this is the tissue that those kidney cells, the mesonephric endothelial cells, will migrate from. And then on top of it here is the gonads that's going to develop into the testes. So we have here an XY mouse or a female mouse that has the SARI gene. In our second sample, we have either an XX or XY mouse, so just a control mouse, that they've stained with LAC-Z, so it's this dark blue color. And then on the far right side, we have an XX gene, or a female XX mouse, or an XY mouse where we've deleted SARI off of that XY chromosome. So it's missing that master switch. Now, what they did is they recombine these in different pairs. So they took that kidney tissue that is stained blue, and they combined it with XY, male, or XX and SARI, female with that SARI gene, gonad tissue. And they saw that if you have SARI, so in this case, the XY male has SARI, or the XX where we've added SARI, we see that migration. And if you were to look at the actual stained experiment, we can physically see that these cells have migrated because they're stained blue to begin with. And so as they move into the gonad tissue, which is normally not blue because we didn't stain it, it starts to take on this blue color. Now, we want to see if this is the case in all mice or only mice that have the SARI gene. So in the control, what they did is they took mice that were female or male, but we got rid of the SARI gene, and we did the same combination. And so we took the gonads and we recombined them with that kidney tissue to ensure, or to see if we can see this migration. And as you can see, because there's no SARI gene in either the female or the males that we deleted the SARI gene, that migration doesn't occur. And so SARI controls this key aspect of migration of these cells to become those blood vessels from the kidney tissue to the mice testes. So as we talked about previously though, SARI is only that master switch, remember? It's only on for a couple hours, then it activates other things like SOX9, and then SOX9 interacted with the beta-catenins to suppress ovary formation. But also, if you remember that figure, at the very top, it interacts with FGF9. And FGF9 will then go back and increase the expression of SOX9 to kind of keep its levels high. And so we know SARI is playing a role, but is it really the end point? Is it the actual protein that's causing this? And what we can do is, or from this experiment, what they did is they looked at FGF9 as a player in this process. So again, we have female mice, and we have female mice or male mice that are lacking or that are stained. And so if we have a female mouse where we combine the genital ridge to the kidney tissue, we see no migration. And usually in our previous experiment we added SARI, but in this case we're going to ignore SARI, and we're just going to take a female mouse and we're going to combine it with a female mouse that is stained, and all we're going to do is add FGF9. And we see that if FGF9 is present in the gonads, then we still get this migration of those cells from the kidney tissue into the gonads to form those blood vessels that are seminal for testes formation. So while SARI is the master switch that allows for this migration to happen, remember SARI activates SOX9, which activates FGF9. So FGF9 is actually the player that is doing the recruitment of these cells to come into the gonads. SARI is just upstream of that. So SARI turns on FGF9, but FGF9 is actually the player that is recruiting these cells to migrate into the gonads. So remember, it's really important that SARI is activated at that perfect time. It's only there for a couple hours, and it has to be active in those couple of hours for this development to properly happen. And so this is kind of a cool experiment where what they did is they took SARI, and instead of having its regular promoter that will naturally have it expressed at the right time, they instead put it under like a heat shock protein or a heat inducible promoter. So this means that they can turn SARI on and off depending on when they heat up the samples or let the samples remain at room temperature. So at 13 hours or 11.1 days after it's supposed to be, or the proper time that it's supposed to be expressed, we see SOX9, which remember SARI turns on SOX9, which is then used, or which is then responsible for the development of testes. So at 11.1 days, they heat up the sample at the proper time, and we see SOX9 is expressed, and that's kind of that key, one of those key genes in testes formation. SCP3 is an ovary developing gene, and normally when SARI is expressed, SOX9 gets expressed, which will repress SCP3. So if it's expressed in the right time, we see that there's very little of this SCP3 expressed, and so we should get testes formation. Now, if we move the SARI expression back a couple hours, so we wait 2 more hours and we heat it up then, we see that in the center of the gonads, we have expression of SARI. However, on the periphery, we're starting to see expression of SCP3. So we're not getting proper wild-type testes formation because we're missing our window. And then if we were to move it back another 3 hours to 11.5 days and then heat up the sample to allow expression of SARI, we see that we've missed our window. And now SOX9 isn't expressed at all, and instead SCP3 is expressed. So even though we have SARI and we expressed it, because we missed our time point that is needed for testes development, we're now seeing a high expression of SCP3, and we're getting ovary formation. And so it's very important, while that SARI is that master switch, it has to be expressed in that proper time. Otherwise, the rest of the development is happening and kind of leaving it behind. And so if at 11.25 days, development is occurring and it doesn't see SARI present, it's just going to go ahead and proceed with ovary development. So now let's look at formation of the external genitalia. And so in males, you have androgens, which are testosterone or dihydrotestosterone, and the receptors for these androgens. And so these are produced by the developing male genitalia and some of those cells surrounding the male genitalia. And if they're present, they block WNT inhibitors. Now, if those WNT inhibitors are blocked, that allows, put a plus here, that allows for the expression of WNT and beta-catenin activity. And once those are expressed and active, they lead to the development of the penis and the scrotum. Now in females, we don't have androgen, so testosterone and dihydrotestosterone aren't present, and we don't have androgen receptors. So there's nothing to block the WNT inhibitors. If there's nothing to block those inhibitors, they will go and they will stop WNT and beta-catenin activity, and it will allow for estrogen and the estrogen receptors to be active, and that will lead to the development of the clitoris and the labia majora. And so it's kind of a simple switch or a simple model here where if you have androgen, you block WNT inhibitors and allow this natural activity of WNT and beta-catenins in males. And if you don't have androgen, you don't block the WNT inhibitors, and that allows estrogen and estrogen receptors to be active and develop female external genitalia. So a key part of that last slide was that to develop into a male and develop male genitalia, you need not only androgen, but you need androgen receptors. So you can send out a message, but you need something to hear that message, right? And so there are some individuals that are androgen insensitive. So they have some sort of mutation or some sort of problem with those receptors for androgen. Even though they're producing testosterone and dihydrotestosterone, they have nothing to receive that message. And so this is an individual here who has an XY karyotype, so they have the Y chromosome of a male, and so they should develop into a male. However, because they don't have those androgen receptors, their external genitalia develops as female genitalia. And so they can still respond to estrogen because you naturally have estrogen receptors, even males do, but because they don't have the androgen male testosterone receptors, they go down that cascade where the WNT inhibitor is still active and they start to develop female external genitalia. However, internally, they lack the Mullerian duct and they still have testes that have developed, and it's only their external genitalia that have developed into female genitalia. So they're internal, they have undescended testes, and they have the Wolffian duct, which becomes the vas deferens. But externally, they have the phenotype of a female because the missing androgen receptor. So testosterone and dihydrotestosterone are important, as we said, for the development of this external male genitalia. And so even though testosterone is always talked about as kind of the masculinity hormone, it's really only expressed in these small contained areas, the epididymis, the vas deferens, the seminal vesicle. And dihydrotestosterone is much more important in early male development. And so if a child has an inability to make dihydrotestosterone, what happens is you get those same phenotypes that we saw in the last slide, where you have non-descended testes and you develop the appearance of a female. However, as the individual reaches puberty, they will start to develop a penis and a scrotum will descend, and then the person that was originally appearing as a female will develop the genitalia of a male. I can only imagine how confusing it must be to, especially before we knew a lot of this early development stuff, to be raised as a girl until puberty and then to develop male genitalia at that stage. But it is something that does happen. In some organisms, like insects, we don't have that normal XY sex determination. Instead, we have XX for females and X0, or just one copy of the X, for males. And so this is where we get this gynandromorph syndrome, or gynandromorph, sorry. And that is an instance where half of the organism is male and the other half is female, kind of seen with our chicken example on the very first slide of the lecture. And so what has happened is that very early in development, at the 1 or 2 cell stage, there is a loss of one of the female chromosomes in one of the cells, and those cells continue to proliferate, and you have then half the organism develop into a female because it has both complements of the 2 X chromosomes required, and one develops into a male. And so you can also see that some of these genes on the X chromosome are non-sex genes, and so we see that we have difference in eye colors and wing size because we've lost the complement of genes required. It's kind of a gene dosage type deal, where the male is where it's missing a chromosome compared to a female that has both complements. And this is also seen in butterflies, as in addition to Drosophila. One very cool sex determination mechanism, I'm very partial. I have 5 pet reptiles at home. Honorary herper, not to the extent of Dr. Shepard, but reptiles are very cool. They have this very cool sex determination system, and we kind of had to cut that chapter because of the amount of lectures we have in a quarter. But they have a temperature-dependent sex determination mechanism. And so depending at what temperature the eggs are incubated at, we have different ratios of males and females. And so in common snapping turtle, we see that if you increase the temperature to about 26 degrees, you end up with a lot more males. But if you were to do about 24.5 or about 27 degrees, then you end up with a 50-50 ratio. In a pond turtle or a painted turtle, at low temperatures, at 26 to 28 degrees, you have all male turtles, and then as you increase the temperature, you get more females. And then alligators, similarly, at about 32.5, 33 degrees, you have all males, but before that and after that, you have kind of this gradient where you can get a 50-50 mixture of males and females. Now this becomes important in an ecological context because of global warming, right? And so as temperatures begin to increase, you can see shifts in this direction of these sex ratios on the curve. So if temperatures rise to the point where common snapping turtles are experiencing only 28 and above, or the pond turtles are only 31 and above, or alligators are 35 and above, then you're only going to have females born. And so at that point, the population is unsustainable, right? Because there's no males to propagate the species. And so in a global context of global warming, this is very important and dangerous for reptiles who require certain temperatures to maintain their species. As you learned earlier in the class, the germ cells are kind of set aside because we need them to stay as pluripotent for the next generation. And so at about 8 days of embryonic development, though, those germ cells start to migrate to the gonads. And here they begin their life as primordial germ cells, PGCs, that are established in the posterior epiblast of the developing embryo over here. And at these 8 days, they start to migrate down. Come on, line, there we go. They start to migrate down to the definitive embryo or endoderm of the embryo. And so this is the location for sexual organ development, but they don't start there. They have to kind of make their way. As these germ cells migrate from the posterior epiblast, they move up the hindgut, the digestive system, and then they move dorsally around here, following these purple arrows, into the genital ridges, which we've shown earlier. This is the tissue that's going to develop into either the mammalian testes and penis, etc., or the female ovaries, uterus, etc. So we can see this migration in actual stained tissue of the mouse. And so here, those primordial germ cells are stained dark. And you can see this is kind of in their migration, partway through their migration, where they would come up the dorsal mesentery or the hindgut and make their way to these two genital ridges. So once these germ cells reach the gonad, we have to start undergoing meiosis to create gametes for the next generation. And this is controlled by retinoic acid, which is secreted by the mesonephros cells and reaches and diffuses to the gonads. And so in females, RA is secreted and reaches the gonad and triggers mitotic initiation via the induction of STRA8. So RA causes the expression of STRA8, which becomes a protein, and then causes these germ cells to start to undergo meiosis. Now, in male cells, there is another gene, nanos2, and what that does is it interacts and it blocks STRA8. So RA still starts the process, but nanos blocks STRA8 and instead causes the germ cells to go down the male pathway as opposed to creating oocytes for the female. However, nanos2 only works in this way after 13.5 days. Before 13.5 days, there is another gene, CYP26B1, that blocks the RA signaling, and so it is antagonistic to RA, so we never get expression or induced expression of STRA8. After 13 days, though, however, this CYP26B1, that expression goes down, and it's not able to do its job anymore blocking STRA8. And that's when nanos comes in and takes over, and instead of blocking RA like this CYP does, it blocks the next thing downstream from that STRA8, and that leads to male induction. So before 13 days, we have one way of blocking female organ generation, and after 13.5 days, when the CYP starts to not be present anymore, nanos takes over and continues to push the gametes to develop into male germ cells. So if we were to look at this from a histology perspective, here we have the genital ridges, and on the left here is where RA is synthesized. So there is a gene, ALDAH1A2. You're not going to have to know that. It's just the RA synthesis enzyme, and you can see where it's active. So it's making RA all along this genital ridge. However, on the right-hand side, remember that CYP26B1, that is what degrades that RA. And so if we look at the left-hand side here, in the male, we see that that enzyme is becoming active and starting to degrade all the RA that's made. So in the male, it's made no matter what, and between male and female, they both make RA. However, in the male, they make something to break it down immediately. And if we were to compare this to the female, like I just said, it's made in both the male and female RAs. And so we can see it being synthesized in the genital ridge here of the female. However, if we were to look at the enzyme that degrades RA, the CYP, we see that it's not present. And so this allows RA then to stimulate the expression of STRA8 and to develop into female genital cells. Okay, let's take a minute to look at how sperm is created in the testes and how it matures. So sperm maturation occurs in the seminiferous tube, which are inside the testes here, as you can see on the slide. And so if we take a cross-section of this tube, we see that the spermatogonia are in blue, which are kind of hard to see on the edge here. And the spermatocytes are lavender, which are the cells that will eventually turn into sperm. And then as the sperm mature, they kind of move to the center of this tubule, and we can see that the mature sperm appear here in yellow. So if we were to look closer at this process, we see kind of the maturation process of these gametes, or these germ cells, into gametes or sperm. So as they mature, we start with this type A spermatogonium, and it will develop or change into a primary spermatocyte, and then further into a secondary spermatocyte, and undergoing the process of meiosis through this, right? Because as these sperm are created, we only want 1/2 the genome, right? We don't, if we have 2 copies of the genome, then every time the organism reproduces, the genome doubles, but we need to be able to combine it with a half genome from the female. Then, from the secondary spermatocyte, it fully matures and moves towards the lumen, towards the inside, because it needs a way of exiting the body, right, for reproductive purposes. So it moves towards the lumen and develops fully into spermatids. As stated in previous slide, the number of eggs, and actually all germ cells in an ovary, decreases over time. It changes over the lifespan. Actually, it decreases after birth. But as you can see, from about 2 months to about 5 months, you actually have an increase in the number of germ cells. So they're reproducing, and you're getting more and more germ cells that start to inhabit, I suppose, the ovary. And then it will decrease to birth, and at birth you have 2 times 10 to the 6th germ cells. But as you age, as a woman ages, the number of germ cells decrease. So you're never going to have as many eggs in your body as you do at birth. And as they're slowly released, they're not replaced. So over time, you get less and less and less of these germ cells, and you get to a point where you will just have no more eggs left. They're not reproduced, and this is generally after the reproductive age is kind of ended and you start to see processes such as menopause, age 50 here. But you have no more eggs, or you have the most amount of eggs at birth, and there'll never be a time in your life that you have more eggs than after you're born. Lastly, I want to talk about nondisjunction at meiosis, and this is very important for all you medical inclined students. This is what leads to trisomies, so Klinefelter's, Turner syndrome, Down syndrome. And this is because of nondisjunction. So nondisjunction is this process where when you line up the two chromosomes, usually you have those spindle fibers come in and pull the two chromosomes apart into the respective daughter cells. And what binds those two chromosomes together is this, this substance called cohesin. And cohesin, it lines the two chromosomes and allows them to stick together. And over time, and this kind of gets everything paired up the right way, and over time, the amount of cohesin in an individual, female for that matter, it decreases. And so it kind of messes with the pairing and pulling apart of the chromosomes during meiosis. And so what will happen is, because this molecule starts to go down, or the amount of it starts to go down with age, you end up with a higher percentage of babies that are born with trisomies. And so as you can see here, the percentage of total pregnancies that that have Down syndrome or Turner syndrome or Klinefelter's is really low, around 2% up until your mid-20s. And then as you get to your mid-30s, it really increases. And this was a concern for my wife and I. So we, as I kind of talked about or alluded to in class, we just had our first child. She was about 34, I think, when she got pregnant. Don't get me in trouble by getting her age wrong. But so she had so for our pregnancy, we had a less than 10% chance. But as she gets older, and we look to have another child, then this chance of a trisomy increases. And there are ways to combat that. It turns out that a lot of the mechanism might not be fully understood, but folic acid has been shown to decrease the number of trisomies in individuals and other birth defects, and how that relates with this cohesin is not fully understood. But proper nutrition is very important during pregnancy to prevent these trisomies from occurring. So that does it for this chapter, and we'll see you chapter 7.